Here we are in prokaryotic cells, section 3.2. And my learning outcome is recognize bacterial flagella in pictures and diagrams. Now, probably most of you have some idea of what flagella are. In human cells, the only cells with flagella are sperm cells. And the flagella allow the cells to move or to swim through an aqueous, a watery environment. So some bacteria have flagella, not all of them, but many different types have flagella. And flagella kind of look like these long extensions. And the structure of a flagella, there's a lot that goes into it. It's really kind of interesting and cool, but we're not going to go into all that detail. I just want you to notice that lots of different bacteria have flagella, although, like I said, not all of them. And they're these long extensions of these specific kind of protein arrangements, make these little tails. But you also notice they can look different. Some bacteria have only one flagella, some have one at each end, some have lots at one end, some have them all over the place. And so here's this is an electron micrograph picture. It's a transmission electron microscope picture. And you can see the flagella all over the outside. There's a light microscope picture with a special stain that shows flagella. Most time you can't see flagella under a light microscope. But in this case, with a special stain, you can. So you can see all those flagella. And then here's one that just has this one flagella off the end. One flagellum is actually the right word, a flagellum, flagella plural. So how do bacteria move using their flagella? Now, there's actually a couple different ways bacteria move, but moving by flagella is the most common way that you're going to see bacteria moving. And when bacteria move, we call this that they're moving by runs and tumbles, and I'll show you what that is. First of all, let's just get some of the vocabulary down. If a bacterium can move on its own, for example, if it has flagella and it's swimming around, you're looking under the microscope and it's moving all over the place, we say it's motile. Motile bacteria are able to move on their own. But there's a lot of bacteria that don't have any way to move. They just sit there, and we call them non-motile. What causes a bacteria to move is it generally needs a reason. And one of the biggest reasons for bacteria to move is they're moving towards or away from food. And food to bacteria are various chemicals. They could be sugars, they could be proteins. They might also move towards an environment that has a more desirable pH. So they might recognize hydrogen ion concentrations, things like that. So bacteria, or most cells in general, have things that we call chemoreceptors. These would be receptors in the membranes of the bacterial cell that can detect certain chemicals. And so some of those chemicals could be food. And so what bacteria generally do is if they're detecting that, oh, things are going pretty well, there's a lot of food around, I can just, go anywhere and there's going to be food, they just kind of go straight. They just keep going on, boop-a-doop-a-doo. They're not looking, they don't have eyes, they can't think, but they're just, as long as the chemical concentration that they like is around, they're just going to keep swimming straight. Now, they swim a little bit too far, and there's not as much food around, and their chemoreceptors don't detect food. Oh, I didn't really define this word chemotaxis. That just means moving towards or away from a certain chemical. So if you have chemoreceptors, this bacteria is moving towards or away from its favorite food. It's using chemotaxis. So as long as things are going good, bacteria are what we call running. They're moving in a straight line. If things aren't going very well, like there's not much food in the environment, the bacteria does a tumble, and they're showing you here how it throws out its flagella and it flips the cell over and twirls around, and then it just starts running again. It starts going in one direction. And the tumble just helped it move somewhere else. Again, because it can't see. It can't just say, oh, I'm going to look over there. I can see there's food over there. It's just going to randomly move in another direction and see if the chemoreceptors detect food. So, and generally, when we look at bacteria under our microscopes, they're just swimming nice and straight, kind of just all over the place, going in straight lines, because there's lots of food in the medium. So they're doing runs. if there was very little food in the medium, or the medium was like the pH was too low or something, you might see the bacteria kind of spinning and tumbling. So runs and tumbles are the way that bacteria normally respond to food, or normally use their chemotaxis. And only motile bacteria can do that, because non-motile bacteria can't move. The other thing I want to talk about in this section is something else on the outside. So we looked at flagella. The other thing I want to look at is something called a glycocalyx. And so it says, describe the structure and function of the two major types of glycocalyces. So glycocalyx is singular, glycocalyces is plural. Describe the structure and the function of two major types of glycocalyces found in some bacteria and the importance of biofilms. Now, a lot of bacteria make a glycocalyx. Glyco kind of sounds like glucose, doesn't it? And it makes you think of carbohydrates. A glyco and calyx means an outer coat. So generally these are layers on the outside of the cell. So in this cartoon, the glycocalyx would be on the outside. And this happens to be the glycocalyx in this case. It's called the capsule. So it's on the very outside of the cell, and it's made mostly of carbohydrates. So a lot of bacteria have these glycocalyces. And there's two major kinds. One is called a capsule. And we're used to that term. We take pills that are in capsule form. And so if you look at this cartoon of a bacterial cell, you'll notice that there's this solid layer on the outside, and it's uniform, it's smooth, and that would be a capsule. Here's some pictures from microscopes, and these would be microscopes that we would use in a live lab. This would be a light microscope. So here's a bacterial cell, and it's got a halo around it, and then it's got some dark color in the background of the slide. And so you see the red bacterial cell. This halo that didn't stain very well is the capsule. So the capsule's about, it surrounds the individual bacterial cells. Turns out the stains we use don't typically stick to carbohydrates very well. They stick to things other than carbohydrates, like proteins. And so you put them in a medium that has a lot of protein in it, and the bacteria can stain and the background can stain, but the capsule doesn't. And you can see that in here too. There's some purple bacteria, and they've got these kind of white or colorless halos around them. Those are capsules. And the reason why some bacteria make capsules is to protect them from the immune system. So the capsule is just carbohydrates, and our immune system doesn't respond really well to carbohydrates. It doesn't make antibodies really vigorously to carbohydrates. So bacteria can kind of hide out from our immune system once it gets inside of our body by having a capsule. And I put on here what is phagocytosis. So hopefully some of you remember that some of our white blood cells are called phagocytes. You might remember the neutrophils and the macrophages are phagocytes. And the phagocytes are white blood cells that actually engulf and surround bacteria. So here's a phagocyte that's coming in to surround that bacterium. But by having a capsule, the phagocyte just doesn't recognize the bacteria as easily as it would if there was no capsule. So when you say, what's the function of a capsule? It helps the bacteria avoid the immune system by avoiding phagocytes. Another question that I wrote in your lecture study guide was some strains of Streptococcus pneumoniae, and that's a common bacteria that causes pneumonia. Some of them make a capsule, others do not. Which are more likely to be pathogenic? Which are more likely to be disease-causing? Are you more likely to cause a disease if you make a capsule or if you don't make a capsule? And I'm going to let you try to figure that out, and the answer's at the end of your study guide. Remember, we said there were two kinds of glycocalyces. So we've got a capsule, and the other kind is called a slime layer. And a slime layer is kind of loosely, it's got a lot of carbohydrate in it, but it's very loosely surrounding the bacterial cells. So here's a special kind of microscopy where you see lots of bacterial cells. They're kind of fluorescent. They're pretty white. And this kind of fogginess around them is what we would call a slime layer. Notice the slime layer looks a lot different than the capsule. The slime layer's surrounding lots of bacteria and blending the bacteria together. Slime layers do all sorts of things that are beneficial to bacteria. I mean, they're kind of wet and moist. They've got, so they can help bacteria retain some water if they're on a surface that's kind of drying out. They can contain nutrients. Remember, carbohydrates are nutrients, so they provide nutrients for bacteria. So if there's not food around right away, they can use their slime layer. You know, they make them when there's a lot of food around, and then they digest them when there's not as much food around. But one of the biggest things that slime layers do is they help bacteria stick together. They help microorganisms stick together, and not just to each other, but they also help them stick to surfaces. And so you probably are familiar with slime layers. I probably think about slime layers every day of my life. I think about them when I'm doing the dishes. You know, when you have a cup, you're washing something, you want to always use a rag or use something that you're scrubbing the surface. And not even just like a dog water bowl. Well, there's not big chunks of things in there, but you'll notice the bowl gets kind of slimy. And that's because there's bacteria in biofilms, bacteria that are making these slime layers. And when we've got lots of bacteria in slime layers, we call them biofilms. So we get these biofilms growing on surfaces. Anywhere there's moisture and there's bacteria, which is almost anywhere that there's moisture, you're going to get these slime layers, bacteria making slime layers and forming biofilms. So you need to scrub bacteria off of surfaces. So where else would you find biofilms? This is a scanning electron microscope picture from the inside of a catheter. Urinary catheters in particular can get a little bit of bacteria in them, and then the bacteria make slime layers, and then they start growing and they stick to the catheter, and before you know it, you've got this biofilm. And now this biofilm is a source of bacteria for infections. So you've got to change the catheter so you don't get these biofilms. Why do we brush our teeth? Why don't we just rinse our mouths, but we actually brush our teeth? Because bacteria in our mouths form biofilms on our teeth. Now, this person looks like they haven't brushed for quite a while. But even if you brushed this morning, you can run your tongue over your teeth and you can feel kind of bumpiness. And those are slime layers. What happens on our teeth is the bacteria that stick to our teeth first are kind of in these slime layers. We can scrub them off with our toothbrush, but if they stay there too long, they actually harden and make plaque. And they stay on our teeth and can cause cavities. So all of that plaque, all those bacteria on our teeth, we need to scrub them off our teeth a couple times a day because those bacteria are making slime layers and forming biofilms. I'm going to stop right there.